Hi, everyone. Thank you, Jason, Aaron, and Polly for organizing this symposium series. My name is Kolade, and I'm really fascinated by the interplay between physics and biology. And one area where this interplay is very important is in cell migration. Cell migration is a fundamental component of the immune response. For example, when T cells exit systemic circulation and then move into peripheral tissues. And in this movie that I'm playing on the left, we see an immune cell that's migrating, but this movie makes it seem like the cell is having an easy time moving around. However, the reality is more similar to what's on the right, where the molecular interactions between the immune cells shown in red and the tissue shown in blue can really restrict the ability of the immune cell to effectively move around. What we've learned from many years of studying cell migration is a conceptual framework that much of the migration machinery is concentrated at the front of the cell, specifically involving this protein called actin, which is labeled in red. This actin protein can form different kinds of physical structures. For example, it can form a dendritic structure called lamellopodia, or it can form linear structures called filopodia. And our current understanding is that much of cell migration is driven by these lamellopodia protrusions. But we realized that previous studies had looked at how cells move around on a glass slide. However, the mechanical properties of glass is very different from those in biological tissues. So then we wondered, how would cells behave if we put them in an environment that's more similar to what they experience in biological tissues? What we found was that if we put cancer cells on a glass slide, they have these beautiful lamellopodia structures that have been previously described in the literature, highlighted in red. However, when we put the same cells in a material system that's more similar to biological tissues, specifically which are compliant and also viscoelastic, we observe the emergence of these really prominent extensions that are extending from the cell surface. We then did a series of immunohistochemistry and biochemical assays to determine that these protrusions are filopodia protrusions. And then more importantly, these filopodia protrusions drive cell migration. We also worked out that the viscoelastic properties of biomaterials regulate filopodia biology, such as their length, the number, and the dynamics of these protrusions, such as their lifetimes and their extension and retraction cycles. So we have a conventional view that lamellopodia drives most of cell migration when cells are on a glass slide. But in more recent work, we demonstrated that filopodia drives cancer cell migration when these cancer cells are interacting with materials that are more similar to biological tissues. Towards the end of this project, we had the SARS-CoV-2 pandemic upon us. And then one thing that I learned is that one of the mechanisms by which these virus infects our cells is by exploiting these filopodia structures. Specifically, the viral particle attaches to the tip of these filopodia protrusions, and in a term called viral surfing in the literature, this particle surfs along the shaft of this filopodia protrusion to infect the cell. So this was really exciting to me because we had already demonstrated in a previous work that the viscoelastic property of materials regulates filopodia dynamics. And so this raises the really intriguing possibility that perhaps viscoelasticity of tissues can also regulate viral and perhaps even bacterial infection. Furthermore, when a T cell forms an immunological synapse with a target cancer cell, these filopodia protrusions are also involved in the very early stages of this process. So this raises a question, does this viscoelastic property also regulates immunological synapse dynamics? So really the point of this slide is to highlight that perhaps our understanding of physics can allow us to probe much more deeply and also broadly into biology, and that these humble filopodia structures play a much broader role in cell biology beyond cancer. And now we have an experimental assay that allows us to modulate and tune the dynamic properties of these structures to elucidate a potential role in viral infection, in cancer, and also in immunology more broadly. One important cell in the immune system is a T cell. And a fundamental challenge that a T cell has to overcome is moving through crowded solid tissues. If we do a quick mental substitution where we replace the T cell with a CAR T cell, and if our goal is to specifically deliver this CAR T cell to a specific anatomical location, this becomes a drug delivery problem. We currently have a good molecular understanding of the interface or synapse between a CAR T cell and a target cancer cell. But one question is, how did the CAR T cell find the targets to begin with, specifically in the context of solid tissues? CAR T cells have to move through solid tissues before they find their targets. And as we look to the future, where we aim to use CAR T cells to solve more complex diseases, we need to have a better molecular understanding of how these CAR T cells interface with the mechanical properties of tissues, and also how these cells interface with other immune cells in the tumor microenvironment, such as macrophage cells. So my future goal going forward is to transform our molecular understanding and engineering of cell interfaces to enable precise delivery of living drugs. And to accomplish this goal, I will pursue an interdisciplinary approach and draw upon themes from mechanobiology, machine learning, cancer models, and cell therapy. And I will start by working with a macrophage T cell as a model system. This is a fascinating system to start with because the physical interaction between macrophages and T cells is important in many disease states, such as in formation of granulomas, in cancer, and also in neurodegenerative diseases. So now we have a system where we can take a macrophage cell, here shown in red, and put it in contact with a T cell shown in green in a three-dimensional viscoelastic matrix, with the goal of elucidating the role of interfacial mechanics in shaping the dynamics of the immunological synapse. This is a really fascinating system to study for a number of reasons. One, there are many scientific questions that one can ask from looking at this just one video. But even beyond that, the surfaces of both of these cells are coated with many kinds of proteins and materials, such as a glycocalyx, that in principle should prevent close contact between cells. Furthermore, in the context of antigen specificity, the dissociation constant between a peptide major histocompatibility protein and a T cell receptor is on the order of micromolar. So to take it all together, we have a system where the transport is limited, the kinetics are low, and there are substantial thermodynamic constraints, but somehow this system still works very effectively. So what I want to understand at a much deeper level is, how does nature take a system that is seemingly on the face of it very inefficient, and then rework that system and transform it into a system that is highly efficient and also very, very effective? This then raises a number of questions that we can address in the future at different length scales. For example, at a biomolecular length scale, what is the role of mechanical forces in regulating the chimeric antigen receptor and cognate ligand interaction time? Does it matter what the nature of these mechanical forces are? Are they shear forces or normal forces? What is the potential role of glycosylation in regulating receptor recycling? At a cellular level, I would like to understand how much time is needed for a CAR T cell and a macrophage cell to transmit information. What exactly is being transmitted? And what is the consequence of this transmission of information on these two cell populations? For example, are there differences in expression of markers like CD25, 69, 62L, TIM3, LAG3, PD1, PDL1, and on and on and on? And at a multicellular level, how effectively can a CAR T cell kill a target cancer cell in tissue mimicking environments? And do tissue mechanical properties promote serial killing? And more importantly, CAR T durability? So this is a really exciting set of questions that I'm excited about for the future. And not only can we ask these questions at different length scales, but we also have a unique opportunity to make basic discoveries that we can then take into translational science. And with that, I would like to thank my PhD advisor, Professor Ovijay Chowdhury, my postdoc mentor, Professor Samir Mitragotri, our funding sources, and I will be happy to connect on social media and answer any questions you may have offline. Thank you for listening.